I'm Dr. Larry Altstiel. I am head of neuroscience clinical research in the neuroscience research units at Pfizer Groton Research Laboratories in Groton, Connecticut. We've talked a lot today about genes involved in Alzheimer's disease, ApoE, APP, PS1, and PS2. Recently, other genes have been discovered to be associated with Alzheimer's disease. Can you tell us what this has taught us about the pathology of AD and what it might mean for treatment?
In addition to APP, presenilin-1, and ApoE4, there are a number of genes that have been associated with increasing the risk for Alzheimer's disease. They seem, however, to be effects that you can only detect when you look at an enormous population. And so far, we can't reduce those to like mutations that we can test for in an individual person. So for APP and PS1 and PS2 and ApoE4, we know exactly where to look in the gene to see where the mistakes might be that cause Alzheimer's disease. For genes like SORL1, which is one of the genes, or clusterin, we don't know exactly where to look. We don't have the precision to know how to do a test that will reduce that to an individual risk. We can only look at that at the population level.
What also these studies may tell us is that Alzheimer's disease is not caused just by a single defect. There will be many factors, many gene abnormalities that, in concert, contribute to the development of the disease. And it's not going to be just a single gene.
It doesn't have to be a gene abnormality. I mean, genes could be important, but it doesn't have to be a gene abnormality. It's not all about genes. There is this part also of science which is called epigenetic, which means it's not the gene that changes the combination of genes, but could be factors that are external to the gene that affect in a different way the genes. So we need to keep our mind open, broad mentality on what we do on these things without focusing excessively. It is important, but it's not the only part of the story.
Yeah, I think the question really prompts a broader question, and that is, as genomic technology gets more and more powerful, and we can identify genes that have tiny effects for this disease or that disease, and we need large populations of people to see these tiny effects. What do we do with that information? Is that actionable? And I think we still don't know. None of us know where this is heading and how we're going to harness this genomics technology.
In terms of what these rarer genes or more genes that we've discovered in populations have told us about the pathology, for every one that's been tested, they seem to dovetail into what we call the amyloid hypothesis. All of these genes, whenever you turn them down in a mouse model, they make the amyloid buildup worse. So they all have so far reinforced the amyloid model of how the disease begins. We don't know exactly how that relates to all forms of common disease, but these genetic risk factors, whenever we've identified them, all seem to, it's possible to fit them into the sort of a unifying picture of the pathology.